Copyright Stanford University, all rights reserved.
Dr. Vagelos is not here, but there are a whole bunch of slides and that I just want to go through. And I think it's better we do that because I want to save the most amount of time for him to give a talk. I want to first thank Dr. Randy Vagelos for inviting his father to show up. And this is a great treat for us. I emailed Roy about 6 or 9 months ago. I didn't really expect what to expect, and he replied back in like 15 minutes and said, "Sure, no problem. I'm happy to visit." So I'm very, very happy that he's here. That's pretty slow for him. Uh, but I think he's talking with Paul Berg, and he'll be here pretty soon. But again, we just want to go through the slides before he shows up, so he's ready to go. And so. Some of you may know this. We have a PCBC meeting in next week, and this is technically it's not open to the public, but I would encourage you guys also to show up. There will probably be about 300 investigators for this meeting, and so these are 10 to 12 different sites across the country that have been funded by this UO1 grant consortium. Looking at iPS cells and ESLs for cardiovascular and renal and other diseases. We also have a talk by Dr. Ken Campbell coming up in a couple weeks, and he's going to talk about sarcomere level function in human heart failure. And then for our next frontier, because next week we don't have the frontier meeting because of the PCBC. For our next frontier, we have Kim Mahaffey, who's going to talk about cardiovascular clinical research. And then this week, actually, we have Phil Yang, who's going to lead the Med 223 talk for the medical students. And then just keep in mind that the talks right now are going to be recorded. And so if you missed the talk, you can go back to the CVI website to take a look at the recording. This is something that we recently set up. It's spearheaded by Holden and Jay Rahadas. We're going to have the clinical biomarker discovery training program. And this is more to teach a lot of our clinicians how do you do biomarker discovery research? How do you take patients' blood, any kind of disease, and run through some of the most fancy biomarker platforms that Stanford faculty have developed. And so Holden's going to talk about multiplexing, and also the nano-immunoassay. And again, this is Holden and Jay would lead this. And then for our retreat, just mark your calendar, December 2nd. Doug Mann, from the Chief of Cardiology, is going to give a talk for our retreat. And travel awards, December 15th, the next deadline. And then manuscript award, January 15th. And then again, look up on the CVI website and also talk to Ingrid. And then the C Grant Awards, I'll send an email out, but this is a list of the C Grant Awards. And because of time, I won't go over the individual awardees. So I want to turn my attention to our guest speaker today. I had a very wonderful chat with Dr. Vagelos. Dr. Vagelos, as many of you know, was the CEO and chairman of Merck for about 10 years before he retired. He obtained his bachelor's degree from UPenn, MD from Columbia, did a surgical residency at MGH, followed by going to the NIH for about 10 years, where he was the section head of biochemistry. And after that award, he went to WashU to be the department chair of biological chemistry. From WashU, he was recruited to Merck as the president of the Merck Research Laboratory. He did that for about 10 years before being promoted to be the chairman and CEO of Merck. And I think everyone would know and agree with me that Roy is basically an icon in the drug industry for what he's done at Merck. Academically, he's a member of the National Academy of Science. He's also on the Institute of Medicine, the American Academy of Arts and Sciences, and the American Philosophical Society. He's a philanthropist, and he has donated a lot of his time and energy to a lot of foundations. He's also was the chairman of the boards at the University of Pennsylvania. Right now he's currently the chairman of the Regeneron Pharmaceuticals, biotech company, who some of you may know is involved in the next generation cholesterol-lowering agent. He's also on the chairman of the board's visitors for Columbia University Medical Center, where he chairs the capital campaign. Very, very important job these days. And so, the list goes on, and I'm just so grateful and happy that Roy agrees to come and give this talk. As you can see by the turnout here, this is going to be a phenomenal opportunity for us. Thank you again.
Working okay? Can you hear me okay? Yeah. Okay. First, as is custom nowadays, one has to admit to bias, and I have people in the audience here who definitely make me biased. We have a son, Randy, who is a cardiologist and doing the things that I thought I was going to do and hoped that I would do. Wasn't so good at it, but he's very good at it. His daughter's here as, I think, his wife, Julie, and my wife, Diana. So this is I've taken time to load the audience. You can't hear. I was just acknowledging the fact that you're here, Julie. Wait a minute. Can you hear this? This is any better? Not better. Can this be turned up a bit? Okay. How's that? Any better? Okay. So I thought I would give you a little bit of a rundown of how I came onto drug discovery as a field and where I started. I started actually went to high school in Rahway, New Jersey, which literally was in the shadow of Merck research laboratories and the headquarters of Merck. Our family had a very small family restaurant in this town, and the entire family worked there. And so I sort of grew up with listening to people from Merck who were largely scientists and engineers. And I was I sort of liked the lifestyle that they had because they loved what they were doing. They were working on vitamins at the time and the very beginnings of antibiotics. And they loved what they did. And so I sort of thought I wanted to be like them. And when I talked to them about going to college, they suggested that I do chemistry. And I went to University of Pennsylvania, as Joe just said, and did chemistry. By the end of the time I graduated, I thought the idea of using chemistry in health was something I would like to do. And so I elected to go to medical school, went to Columbia Medical School, and was immediately turned off by the fact that the medical school was largely, in the first year was anatomy, and I have almost zero memory, and so I almost died during the first year, and had to be resuscitated over the Christmas holiday. Went back to medical school and finished, but by the time I finished medical school at Columbia, I was absolutely convinced that I wanted to do clinical medicine because taking care of patients had become very important to me. I went from Columbia to the Mass General Hospital in Boston, where for 2 years I was in the Department of Medicine, and there was absolutely sure I wanted to do clinical medicine because around me I had these fabulous people, both on the faculty and the fellows in the on the house staff, the residents, were just fabulous people. And you would recognize many of the names who were there at the time, simply because they did so much after that. But that was the time of the doctor draft. And as I was finishing, I was invited to the NIH, where I met Earl Statman. The deal going to NIH was it fulfilled the doctor draft, 2 years of government service. I would go there, take care of patients half-time, and the other half of the time, I would be able to do any kind of research. I met Earl Statman for the first time, and Earl was a very exciting biochemist there at NIH. And I was told by a person at the MGH, Paul Zamesnik, that if I were to go to NIH, I should work with Earl Statman. So I sat with Earl. Earl spoke very softly. I could barely hear him. But as he spoke about his research, he became more and more excited. Now, as he spoke so softly and he used words that were beyond me, I didn't really follow what he was saying, but he was so excited that by the end of our discussion, I was excited. Just sort of, it permeated the place. And so I asked to work, whether I could work with him. And he looked at me and he said, "I've never worked with an MD." So I looked at him and I said, "I've never worked with a PhD." And so we gave it a try. At any rate, I worked with Earl pretty much as an apprentice. I worked on a bench right next to his, and he showed me everything. I was an idiot. As I started, every instrument he taught me. He lent me his textbook, which he used when he was a PhD at Berkeley. And I worked my way through the book during the evenings, and during the day, I would do experiments that he would explain. And then, at any rate, I stayed, instead of 2 years that I owed to NIH, he induced me to stay longer. I stayed for 10. And during that time, I learned biochemistry to the degree that I was okay. And I worked on fatty acid biosynthesis. I loved what I was doing in pure basic research. We worked out the entire cascade of reactions that explain how fatty acids are made in all biological systems and the central roles of acyl carrier protein, etc. And by the end of the 10 years, I had grown away from any of the clinical work that I had been doing at the start. So my job was to take care of heart patients, but at the end, I did only biochemistry because I was sucked into the research through intense interest in what was happening in the laboratory. At the end of that time, I shifted over to Washington University, where I was in the biochemistry department. I was chair of biochemistry, and there continued my research work, but started teaching. And I taught medical students, graduate students, undergrads, and became very much involved in a huge research effort. Again, fatty acids, complex lipids, ultimately cholesterol. And so I covered the whole field and was very happy, would have been happy my entire life, except by that time, I had a couple of calls to be considered as a candidate for a deanship at two medical schools, the University of Pennsylvania, my old school, and the University of Chicago. It was a frightening experience. I thought that I was now being considered as a dean, and that really scared me. And then I had an invitation. I didn't mention that I spent one summer as an intern at Merck in my hometown while I was a medical student. And it was a boring couple of months in screening for antibiotics, and I had no interest in ever thinking of going back. But I had a call from the head of the person who led the laboratory that I worked in, who had become a vice president of research. He called and asked whether I would visit Merck and consider coming back as head of the research laboratories. I said I had no interest, and I had not thought about drug discovery. He said, "Well, why don't you visit?" So I did. I visited the labs, and what I discovered was that Merck research, as well as much of the industry, was using for drug discovery animal models of disease. So they would take an animal and make it, give it high blood pressure, an infection, kidney disease, whatever. And then the chemists would make what they thought would be interesting chemicals, which would be fed or injected into the animals. That was their mechanism, their strategy for drug discovery. And I thought, my God, think of what can do if one used biochemistry for drug discovery. And I said, gee, this might be interesting. I went back, talked with Diana, said this could be interesting. And I went back again and talked with them. And I met with the CEO, Henry Gadsden. And he said, we'd like you to take this job. And I said, Henry, if I came to Merck, I would change this entire strategy for drug discovery. Is that okay with you? And he said, if we weren't talking about something big, big change, we wouldn't be talking with you. Okay? So what would happen if a company got into trouble and the sales were not doing what you expected? He said, well, we'd cut back on all the staff groups. We'd squeeze down on manufacturing. Sales and marketing would have to be shrunken down. But we would never cut back on research as long as I could lead this place, because research is our future. And that was a pretty interesting response to me. And so I agreed to join Merck in 1975. When I went in, the idea was to use the strategy of focusing on molecules that might be involved in disease. And the molecules would be the ones I understood: enzymes, receptors, ion channels. And so it would be molecular targeting rather than animals. And I spent the first year in the laboratory going around talking to various people who all had great expertise in what they were doing in the pharmacology, physiology, essentially no biochemistry. And they had wonderful chemistry, very smart chemists. And so we went around and tried to pick biochemical targets for drug discovery. And I'd like to tell you about a few of them. And for those of you who know some of this history, I'll bring you up to date on where those fields are. The first one we dealt with immediately was hypercholesterolemia and the cholesterol hypothesis. The hypothesis of that, this is 1975, was based on the observations of the fact that coronary arteries, after a person has a heart attack, those plaques are filled with cholesterol. So that was one clue. Another one was the fact that you could cause animals to have heart attacks and have plaques in their arteries by feeding them. These are rabbit experiments. Thirdly, there was the epidemiology worldwide, and that is, if you look at incidence of death from heart attack in Japan, it's very, very low, and the levels of blood cholesterol are also exceedingly low, whereas people in Finland have an incidence of death from heart attack that's 14 times higher. And their blood cholesterol levels were sky high. And if you look across the globe, there's a straight line relationship between death from heart attack and blood cholesterol. And so those were the clues. But the most important thing was understanding how the body makes cholesterol. And Lenin in Munich and Bloch at Harvard worked out the entire pathway of cholesterol biosynthesis. And Lenin, in his Nobel Prize speech, his lecture, pointed out that in the biosynthetic, 20 enzymes that are required for cholesterol biosynthesis, one is rate limiting. And that was said in 1963. In 1975, there was very little action in this field that I knew of. So we decided that that should be the place, if we're going to try to block the whole system, to go for the rate-limiting enzyme. And so we were to do that, and Al Alberts, who had come from Washington University, and we had worked together for many years, agreed to take on that project. Just as we were starting up, we heard from a publication, Endo in Japan had discovered an inhibitor of the enzyme, this rate-limiting enzyme, which is HMG-CoA reductase, the enzyme which converts hydroxymethylglutarate coenzyme A to methylonic acid. Endo reported an inhibitor called compactin, which was a reversible competitive inhibitor of this enzyme. And he isolated this compound from a soil microorganism, so it was a natural product. And it did everything we were going to be looking for. So right at the start, we knew we were behind. And nevertheless, Al, I said, Al, you know, we might catch up. And so Al took on the challenge, and sure enough, the Japanese group, this was Sankyo Company, Endo was in Sankyo Company, did all the animal studies and then raced into the clinic, and we're moving very rapidly to show that this compactin by blocking HMG-CoA reductase actually would reduce blood cholesterol, and they were very close to showing that when Al came up with lovastatin, another product of a microbial, a soil microorganism, natural product. Rather similar to compactin in structure, but different. And so Merck now had an inhibitor, looked very similar to compactin in what it could do, and did the animal studies and went into the clinic. And so we're catching up. When one day we were called and told by someone in Japan that all the studies of compactin had been stopped. And the rumor was stopped because it was found to cause tumors in animals and dogs. Immediately, since we had an inhibitor of the same enzyme, we considered the possibility that if this rumor was true, and we could not get it confirmed, because when I called the laboratory and talked to the head of research and asked about details of what happened to compactin, they said it was an industry secret, and they could tell us nothing. And so we were left with the rumor that there were tumors in dogs, and they had stopped the research, their clinical research. And so at Merck, we stopped the clinical research with lovastatin. And we were out of the clinic for about 2 years. We waited to do animal studies to demonstrate that lovastatin, in fact, did not cause tumors. And we went quickly into the clinic, and with FDA approval, of course, and doctors who had patients with very high blood cholesterol who were really pressing to find out what something that blocked HMG-CoA reductase would do. We went into the clinic and in large studies demonstrated in patients who were already at high risk. The FDA said, look, we don't know that this is going to work, so you've got to be high risk. So these were patients who had coronary heart disease and high blood cholesterol. And they were tested, and the lovastatin caused a nice reduction in cholesterol, LDL cholesterol, small increase in HDL cholesterol, and it was safe in the clinic and approved as the first statin in 1987 anywhere in the world. Well, some doctors liked that idea, liked the idea that they could reduce blood cholesterol, but others did not. They thought, so what? You're reducing blood cholesterol. How do you know you're doing any good for the patient? And Merck, on the other hand, had brought along, while waiting for the toxicology, brought along a second one, which was simvastatin. And simvastatin was the second statin in the world that really buzzed through the clinic. And we did the first outcome study, large outcome study with a statin. That was simvastatin. Some of the older folks here would remember the 4S study. It took again, this is simvastatin, more potent than lovastatin, a specific competitive inhibitor of HMG-CoA reductase, was taken into an outcome study that studied 4,444 patients. All of them had some sign of coronary heart disease and high blood cholesterol. Half of them were put on the drug, and the other half were put on a placebo. And this was an event-driven trial, clinical trial. It was double-blind. At the end of 4.5 years, the trial was stopped. And they looked at the results, and what they found was that those people who were on drug as compared to placebo had a reduction in death from any cause of 30%, reduction in death from heart attacks, 42%, a reduction of strokes, 30%. And these, of course, were all highly statistically significant. And that one experiment changed the cholesterol hypothesis to fact. And all the people who did not believe before believed that cholesterol, LDL cholesterol lowering, was important. And that essentially started a revolution in treatment of coronary heart disease and heart disease. That was all happened in the early 1990s, and then there was a whole stream of new statins. One followed the other, and every new statin was able to reduce cholesterol and LDL cholesterol a bit more than the previous one. That allowed them to come onto the market, of course. And so there was a whole stream of these things, and over the time, I think most people would agree that reducing LDL cholesterol is a good thing. Now, in the meantime, after the discovery of the statins, the true mechanism of reduction of LDL cholesterol was discovered really by Brown and Goldstein, who had discovered the LDL receptor in liver cells. And for those, the LDL, we have the thing circulating, the cholesterol in their lipoproteins circulating in the bloodstream. The level is regulated by a receptor on the liver, and that receptor has a high affinity for the LDL cholesterol. And when it traps it, it sucks it into the liver, interiorizes the LDL and degrades it. And so the receptors actually suck the LDL cholesterol out of the bloodstream, thereby reducing the blood cholesterol. And the mechanism is that when the liver cell senses a reduction in biosynthesis of cholesterol, when you use a statin that blocks the synthesis, it pops up new receptors and to bring in cholesterol, and thereby reduces the LDL cholesterol. So in fact, that's the mechanism that is reducing LDL cholesterol in the bloodstream. So that whole theme developed over the years. The question was then, is there a way to further reduce blood cholesterol? And because although some people can get down into a normal rate, whatever the normal range is, get a radically reduced LDL cholesterol, which is, everyone believes, is going to be a very high risk factor for coronary heart disease. Some people could not get down far enough, and some people could not tolerate statins. And is there another way to do it? And so over the years, people looked for that. And the next major developments came from an unrelated field, and that is some people up in Montreal, headed by Nabil Sida, were working on proteins circulating in the blood, pro-protein convertases, a group of enzymes. And in 2003, he announced that there was a particular pro-protein convertase called PCSK9, which was very interesting in that he identified the gene that was coding for this protein, but he didn't know what it did. In France, a woman by the name of Catherine Boileau was doing genome studies and looking at people with very high blood cholesterol. And she was trying to figure out how these people who were very hypercholesterolemic, very few but very high, and she was honing in on a gene that was mutated in these patients. And the two of them got together, SIDA in Montreal and Boileau in France, and they concluded that the PCSK9 gene that was being studied by SIDA was the gene that was mutated in these patients with very high blood cholesterol, LDL cholesterol. And so now Helen Hobbs, geneticist, human geneticist in Dallas, heard about this. And what the report was, here a mutated gene was causing an increase in PCSK9, which was causing an increase in LDL cholesterol. So it was a mutation that was increasing the amount of an enzyme. Hobbs said if there's a mutation that increases a protein, there must be a mutation that will eliminate the function of that gene product. And there was a very large population study in Dallas where she was focusing on LDL cholesterol. And so she had several thousand people. She was looking at genomics of that and doing looking at exactly what was characterizing these patients. And she looked for people who had extremely low blood LDL. And there were a group, and they were mutated in the same gene as the ones that had a mutation to cause very high low-density lipoprotein. These had exceedingly low. Those that had, it was a mutation in PCSK9 gene, and if there were 2 copies of this mutated gene, some of these patients had levels of LDL down at 15 with a normal of 100. And these patients with exceedingly low LDLs had a very low incidence of coronary events. Of course, small numbers, but this was the beginning of the understanding of another mechanism to reduce LDL cholesterol. And Hobbs, being in Dallas, of course, with Brown and Goldstein, recognized that the protein PCSK9 would bind to the LDL receptor on the liver, and that would cause the LDL receptor would pull in the PCSK9 and inactivate it, and so it became clear that what you wanted to do was to reduce the PCSK9, and so several laboratories, of course, jumped on to trying to do this, and many have made monoclonal antibodies to inactivate PCSK9 to see whether, in fact, this would be a new mechanism for reducing blood cholesterol. And sure enough, the 2 laboratories that are in the lead at this time are Amgen and Regeneron. And each of them has a monoclonal antibody that, when injected into patients who are either already maxed out with statins or without any statins at all, can reduce LDL cholesterol dramatically. And you see, for instance, if you take a patient with high cholesterol and LDL cholesterol and inject them with this monoclonal antibody, they get a further reduction of 50%. So this is something that's very exciting for cardiovascular disease. Both companies have finished their clinical phase 3 studies and either have or are about to file for approval. And so one could predict that if approved by the FDA, the drugs would be on the market next year. But outcome studies are being done now, will not be done for a couple of years, but these outcome studies will take patients with high cholesterol, LDL cholesterol, reduce their blood cholesterol as far as they get with the maximum dose of a statin, since that is the standard of care, and then half of those will be put on the antibody and the other half on a placebo to look at outcomes. And if one believes the LDL story that I've been telling you, this would be the, one can anticipate a reduction in death in those patients who have further reductions. Now, so that was one target that we used. Another one that the idea was to find places. Oh, I should point out how important the information that was coming from universities was in this. Understanding cholesterol biosynthesis, the epidemiology, all the stuff that I explained earlier, and the interaction between the industry laboratories and based completely on what had happened in academic centers and how important that is. Second example is the drug experience that we had with the problem of benign prostate enlargement. And this was a study that recognized that older males, my age and even younger, get enlarged prostates that ultimately can put pressure on the urethra and block the flow of urine. And so there was an ongoing project in the laboratory when I got there, but they were using the old-fashioned approach. We became immediately aware of work of Gene Wilson at Dallas and Julianne Imperato McGinley at Cornell, who were studying pseudohermaphroditism in males. And there were a group of pseudohermaphrodites in the Dominican Republic. And these folks were infants when they were born. These males had very unusual external genitalia. The testes did not come down, they did not descend. The penis had not formed completely, and so they had ambiguous looking external genitalia, which were thought to be female at birth. So these pseudohermaphrodites were females at birth. They were brought up as little girls and dressed as girls until they went through puberty. When they went through puberty, they masculinized. They got beards, they became muscular, they had a male sex drive. And so this syndrome was being studied by Wilson and Julianne McGinley. What they discovered was that they studied the two hormones that they thought might be related, the masculinizing hormone testosterone and its metabolite dihydrotestosterone. And what they found was that there was, in those people with this syndrome, the levels of dihydrotestosterone were extremely low. And so they looked in the bloodstream. There was a reduction of about 70% of dihydrotestosterone. Testosterone is reduced to dihydrotestosterone by a single enzyme. In the prostate of these people, there's almost no dihydrotestosterone. It was reduced 95%. And so the observation of these people was that they were male when they became mature. They had no male pattern baldness. They had tiny, tiny prostates that never grew, and they never developed prostate cancer. And so we had the idea that if we were to block the formation of dihydrotestosterone in a developed male, an older male, that we might be able to stop overgrowth of the prostate, because these people had tiny prostates, and who knows what else. And so Gary Rasmussen took on the project of designing an inhibitor of the 5-alpha reductase, the enzyme that converts testosterone to dihydrotestosterone. And within a couple of years, he designed such a safe inhibitor. It was called finasteride, and it was put into animals initially, shown to shrink the large prostates of old dogs, and then it went into humans. And ultimately, the outcome study, which I'll tell you quickly, was to take older males with large prostates, and it was a 3,000 patient study, double-blind again, using this inhibitor for the conversion of testosterone to dihydrotestosterone. You give the drug to the patients, and the levels of dihydrotestosterone drop 70%, similar to what was found in these people, and over time the prostate shrank about 25%, and the symptoms resolved to a large degree. And so it was a terrifically interesting thing. It caused some, and it did cause some sexual dysfunction in 8% of patients, but it was reversible. The interesting thing was that in long-term outcome studies, it reduced the need for surgery for relief of the pressure from the prostate, 55% less surgery over a 5-year period. And so it was very important, a very important drug. Now, it went on the market, and then we had the idea, if this is a trophic hormone that we're reducing, perhaps it would also, reducing the amount of dihydrotestosterone would also reduce the incidence of prostate cancer. The NCI was convinced that this was a good idea, and they undertook a very large-scale study to test whether reducing dihydrotestosterone over a long period would block the formation of cancer. And they undertook 18,000 patients, started older males, all normal prostates. Half of them put on finasteride, which is called Proscar, the other half on placebo. And again, an event-driven trial, double-blind. And at the end of 7 years, they stopped the trial. And what they found, because there was a 25% reduction in cancers. And so that was an amazingly positive and exciting for the moment. But then it was discovered that when they looked at the level of aggressiveness of the remaining tumors that were found in the two groups, that there was a higher incidence of more aggressive tumors in those patients who were in the drug group. And so that was, couldn't understand that. It was actually to be expected, because you would expect to reduce the cancers that are sensitive, hormone dependent, and you would not expect to reduce the other cancers. And so that was an expected thing. The FDA never approved it for use in preventing cancers because of that fact. Long-term follow-up of this population has demonstrated that, in fact, there is no increase in aggressiveness, and death from prostate disease and prostate cancer complications is the same in the two groups. So you really have done nothing to increase the aggressiveness. What you've done is eliminate the other tumor. You've reduced the other tumors. And so that is something that remains to be done. So that was another incidence, another example of using an enzyme blocker. Another brief story, and that is the use of biochemistry in developing a vaccine. In this case, the vaccine for hepatitis B. And hepatitis B is a viral infection that you know was very dangerous. It was very high incidence in patients, in people who are either IV drug users or gay males. And these people would go on. Many of them would resolve their viral infection, but a small number would go on to either have chronic hepatitis, cirrhosis, or liver cancer. And there was, when these studies began, there was no treatment for that and no vaccine. Barry Blumberg was the person who first identified, again, academically, identified a protein in the blood of people in Australian Aborigines. And this protein was found when he was doing epidemiological studies looking for markers of disease in gel electrophoresis. And he found this unusual protein, which he termed the Australia antigen. Came back to the United States and showed that this antigen this protein would react with an antibody in patients who had had serum hepatitis. And over a number of years, Barry Blumberg discovered and characterized the hepatitis B virus and characterized this band as a surface antigen. Well, surface antigen he proposed and demonstrated that it was antigenic in mice and wrote a patent that it could be the basis of a vaccine. Now, Maurice Hilleman at Merck undertook to actually isolate this in large amounts and try to make it a vaccine for humans. And Hilleman did that. He collected blood in those people who had the antigen, who were intravenous drug users and gay males, collected large amounts, purified it to homogeneity as far as he could tell, inactivated, but went through several treatments that would kill any viruses in the product, and he made a vaccine, converted this into a vaccine, which went into human trials and was shown to be protective and put on the market in 1981. Well, 1981 was the beginning of AIDS, and people were very concerned that anything made from blood from people who are dying of AIDS might be contaminated. The cause of AIDS wasn't known at the time, but people were very suspicious of the vaccine. Fortunately, we had gotten together with Bill Rutter in 1978 to clone the gene from the virus that makes the surface antigen, which was the basis for the vaccine. And Bill Rutter cloned the gene, and he and then Ben Hall got together and put the gene into baker's yeast. And very shortly, baker's yeast was a source now of an antigen, a protein that could be produced in large scale and was shown to be just as effective a vaccine as the one that was isolated from blood, and it was over 95% effective. The Chinese, it was approved in 1984 for use in the United States. It was initially for high-risk people, but soon it was used in all children. Chinese came to us about that time, the late 1980s, and asked whether they couldn't have the vaccine for a very low price, and we tried to sell it to them at cost. And they said that was too much money. And so they talked with us for a long time. We agreed to give them the technology for just paying for the training of their people, which was $7 million over 2 years. And they undertook this to come to Merck and to learn to make the hepatitis B vaccine, because 9% of the Chinese population are carriers of the virus, and our hepatitis B virus is one of the top causes of illness and death in Japan, was at that time. We found that the vaccine, the transmission in China is from mothers to newborns within the first 48 hours of life. And so as they got going, the Chinese learned to do it. Chinese engineers and scientists were trained at Merck, and then they and the Merck engineers went to Beijing and Shenzhen and built 2 plants, and that with enough capacity to immunize all the newborns in China, 20 million a year. And this program was started in 1994, and a decade later, the Chinese were immunizing 95% of their newborns. And this was accomplished with the Merck vaccine, and Merck made not a penny from that program, but they have very good friends in China now. So the Chinese have a lot of money and could afford anything, including buying Merck. So this is one last story, and that is the story of river blindness, which is just another example of what a company could do if it is able to use research to develop important drugs. And that is in 1975, when the early product candidates came out of the research lab was a drug called ivermectin that killed, in an incredible way, parasites. And the mechanism, again, the mechanism discovered at university was that this drug was able to go to the glutamate-gated channel for chloride. It was the chloride channel, which would be inhibited by ivermectin, this macrolide that was discovered at Merck. And when you did that, it became a nerve poison. So it was a nerve poison in the nervous system of parasites, gastrointestinal parasites. And it was very exciting because it was so potent. It could be used in cattle, horses, sheep, pigs, everybody who was having a problem with parasites, but not humans, because humans have tapeworms and hookworms, which are not very sensitive to ivermectin. And so it was put on the shelf and used for animals until one of our infectious disease doctors, Mohammed Aziz, came and told me about river blindness. The river blindness was a parasitic disease in largely in sub-Saharan Africa. It was infecting, the cause is a parasite Onchocerca volvulus. It was infecting 18 million people. Over 100 million were at risk. So the proposal by Mohammed Aziz was that he take the drug and try it in some people with this disease. And as he did, and he went to Dakar in Senegal, found people with this disease that had obvious microfilaria in their blood, in their skin, took a pinch of skin, counted the worms from these Onchocerca volvulus microfilaria, and found that they averaged about 25 microfilaria per pinch. He then gave one tablet of ivermectin, came back in a month, did a pinch of skin again, and at that point found that there were none, zero. We tried to get the World Health Organization to come in and help us with this project, but they came in, reviewed the data. They didn't believe that it could be that effective. And besides, they had bought planes to kill the flies that were transmitting this thing. And so they walked away, and Merck started a program in Africa among the poorest people in the world. And the program was to go down there, identify people with river blindness. The disease is carried on by this parasite, would get into the skin. The microfilaria would crawl through the skin, cause terrible itching. They'd get into the eyes and cause blindness. And so the experiment in this case was to start with a large group of people who had obvious disease, give them each, take a skin snip, count the worms, and then give them each one tablet, and then come back at 3 months, 6 months, 9 months, a year, and take a pinch and count again. And as they did that, they realized they had none at 3 months, none at 6, 9, 12 months. They had a few. So it became apparent that they had a drug that could be used as one tablet once a year and would block the blinding of millions of people. And so the question was, these were among the poorest people in the world. So we carried out the entire program. At the end of that time, 1987, all the data were in. The drug was safe. It could block river blindness and the itching, but there was no ability to get the drug to the people. People couldn't afford it at any price. Their governments did not think blindness was that important because these people were starving anyway. So we went to the U.S. government. We talked to them to see whether they would do this. And I met with the chief of staff of President Reagan and I met with the Deputy Secretary of State, and each of them said, yeah, this is terrific. This is really, but we'll plant the American flag all over Africa. I said, yeah. and as we walked out of their office, said, Dr. Vagelos, this is such a fabulous thing we could do, but unfortunately we don't have any money. And I said, we're talking about $2 million to start this program. He said, you don't understand. It's not in the budget. And so we're in a tight spot because the word had leaked out that this drug was working in these poor people. There was an article, it was in the New York Times magazine section, a photograph on the front page showing blind people of Africa. And the story was about Merck having this super drug. And what was Merck going to do? Well, suddenly we had a call from France. The French authorities had reviewed our registration filing and found that it was appropriate for approval. But we didn't have a decision as to what we were going to do with it. So we quickly got the executive group together and decided this research group could not be left hanging. And so we had a press conference that Monday, right after the weekend, and announced to the world that Merck would contribute the drug free to anyone in the world who needed it for as long as it was needed. That was done in 1987. In the interim, I, of course, had to step down when I was 65, which was about 20 years ago. But the program has continued over the years. The World Bank, the company contributes the drug free, but the distribution into the bush where it's needed in Africa and Latin America is helped by the World Bank. The World Health Organization has gotten back into it, a number of foundations, and the Jimmy Carter Center. Jimmy Carter became one of the chief salespeople for getting this drug distributed. The program has increased, each year has increased in the number of people who were treated. And I forgot to tell you that about 5 years after we started distributing, we found that the drug also works in lymphatic filariasis, which can cause elephantiasis. And so the population target went from 100 million to 1 billion. Well, the program reviewed it in year 25, which was 2012. Merck contributed drug that year to treat free 200 million people. Unbelievable number. 200 million people will not get river blindness or elephantiasis, lymphatic filariasis, on the basis of a free drug. The target is to eradicate the disease. And so it's an incredible program. Well, those are 4 examples that I've used where biochemistry and understanding science can do great things in drug discovery. And I will tell you that looking forward, it's even more exciting as you look down what is happening. New cancer drugs that are being developed, and one was just approved in these immuno-oncology drugs, which are game changers. In every one of these instances, there is basic research that is coming from universities and academic institutions, which is absolutely required to be able to do the translational research. I used to call it applied research. And so my concern, a concern that is quite troubling, is that there is a push by the National Institutes of Health to get people in academia to do more translational research. And what we lack is more understanding of biology and pathophysiology, pathogenesis of disease, cause of disease, and the molecules to